This is Dr. Morris, and I'm continuing the lecture series in microbiology with Chapter 4, Part A. This chapter will describe the functional anatomy of the prokaryote and the eukaryote cell. Prokaryotic cells, when comparing the prokaryotic and the eukaryotic cells, prokaryotic comes from the Greek word pre-nucleus. Eukaryote comes from the Greek word for true nucleus. Distinguishing characteristics of prokaryotic or pre-nucleus cells are genetic material, DNA. This is one circular chromosome, and it's not enclosed within a membrane, lacking other membranes and enclosed organelles. Now, those of you who have had AMP1 and AMP2, if you remember back to Anatomy and Physiology 1 under cellular anatomy, we talked about the cell membrane of the human cell, and then many of the organelles inside of the cell also have a cell covering as well. As you can see, when we talk specifically about looking at the characteristics of the prokaryotic cell, we see it lacks other membrane enclosed organelles. There's no histone proteins associated with the DNA. Cell wall almost always contains a peptoglycan, and they usually divide by binary fission. Distinguishing characteristics of eukaryotic or true nucleus cells: the nucleus is bound by a membrane. DNA and chromosomes consistently associated with proteins are called histones and non-histones. Possesses mitotic apparatus. It possesses membrane enclosed organelles such as mitochondria, endoplasmic reticulum, and sometimes chloroplast. As you can see, the similarity between the eukaryotic or the true nucleus cells are much more similar to what we have studied previously for the human cells. This is a table out of your textbook that gives a comparison, again looking at the chromosomal pattern, looking at the histones or no histone presence. As you look at prokaryotics versus eukaryotics, no organelles and organelles, peptidoglycan cell wall, polysaccharide cell wall, and then binary fission for prokaryotes and mitotic spindle for eukaryotes. Should be familiar with these characteristics as displayed here on this slide. Prokaryotic cells: size, shape, arrangement of bacterial cells. Bacterial range in the size from 0.2 to 2.0 micrometers in diameter, 2 to 8 micrometers in length. Bacterial shape are spherical. Cocci are berry-shaped. Rod-shaped are bacillus, meaning like a staff, and then the spiral shape. Diplococci form pairs. Streptococci form chains. Tetrad divides into two planes, forming groups of four. Sarcini divide in three regular planes and form tube or cube-like patterns. Staphylococcus divides in irregular random planes and form grape-like clusters. Most bacilli are single rods, but they appear in pairs, random planes, and form grape-like clusters. Most bacilli are single cells, but they can appear in pairs, diplobacillus, or in chains, streptobacillus. And then coccobacilli are ovals. Vibrio, and you'll hear this name described when we talk about some microorganisms in the future, such as in cholera, slightly curved, comma-shaped rods are also included among spiral bacteria. Spirilla have a helical corkscrew shape and are motile by means of a flagella, and we talked about flagella in the last lecture. Spirochetes are shaped like spirilla but have axial filaments for motility, and pleomorphic bacteria have irregular morphology. If they maintain a single shape, they are monomorphic. These are some common shapes that we see in the environment, and if we look at these pictures microscopically, this is what the organisms will look at. Two cocci joined together, diplococci, cocci in chains, streptococci, bacillus, spirilla. All of these are very common. Here are two rods in shape. Usual shape: star-shaped stela, square, helicoarcula. Most bacteria are monomorphic. Few are pleomorphic. And again, these pictures are taken from your textbook. And again, just the arrangement as I previously described: diplococci and diplobacillus, clusters, staphylococcus, chains, streptococcus or streptobacillus. The general term of substances around bacteria cells are called the glycocolloids, which is usually a polysaccharide or polypeptide or both. Again, those of you who have had AMP1 and 2, if you think back to AMP1, we talked about the glycocolloids that covers the cell membrane in the human cells, important in recognition. As you can see, similarly, we see this in the cell walls of bacteria as well. Organized and slightly attached is called a capsule. If unorganized or loosely attached, the glycocolloids is called a slime layer. If made of sugar, it is called an extracellular polysaccharide. The glycocolloids aids in the attachment of surfaces of the capsule and contributes to the pathogenicity by protecting from phagocytosis, an important part of the body's defense. Outside the cell wall is where the glycocolloids will be present. It's usually sticky. The capsule is neatly organized, or a slime layer that's unorganized and loose. Extracellular polysaccharide allows the cell to attach. The capsule prevents phagocytosis. Again, phagocytosis being an important mechanism in the human body to be able to engulf foreign invaders such as bacteria and to rid them of the body. With the capsule, as you can see, capsules are typically resistant to the process of phagocytosis. The flagella, as we've alluded to earlier, filaments that are composed of protein, flagellin. The base of the flagella filament widens to a hook. Attached to the hook is the basal body, or a rod with rings, which anchors the flagellum to the cell wall and the plasma membrane. The basal body of the gram-negative bacteria is anchored to the cell wall and the plasma membrane. The basal body in the gram-positive bacteria, it is anchored only at the plasma membrane. Flagella, when present, is arranged in one of four basic ways: monotrichous, a single polar flagellum; lophotrichous, two or more polar flagellum at one end of the cell; amphitrichous, a single flagellum at each end of the cell; and peritrichous, flagella distributed over the cell. You should be familiar with these various types of arrangements, the basic arrangements of the flagella: monotrichous, lophotrichous, amphitrichous, and peritrichous. Flagella may spin clockwise or counterclockwise, producing directional movement. Run or swim is the other terms that's used, or random changes in directions referred to as tumbles. You have illustrations of this, and you have a dynamic media file as a part of your lab that gives you a visualization of these bacteria and how they would look under the microscope. Movement to or from the stimulus is called taxis. The stimuli may be chemical or chemotaxis, or light phototaxis. A flagella protein, H antigen, is useful in helping to distinguish between serovariants or variations within species. Let me give you an example. The H5 virus of the flu virus, commonly referred to as the avian flu, the H is the antigen portion, and it helps scientists and microbiologists to be able to identify one species from another. Thus, it distinguishes the serovirus of various infectious organisms. In that example, that would be an example of a virus. Flagella exist outside the cell wall, made of chains of flagellin. They attach to the protein by a hook, anchored to the wall and membrane by a basal body. This gives you an illustration of the dynamics of the flagella. These are the flagella arrangements: the monotrichous, the amphitrichous, the lophotrichous, and the peritrichous. Should be familiar with these various structures. The motile cells of the flagella rotate flagella to run or tumble, moves toward or away from a stimuli. That's referred to as taxis. Flagella proteins are referred to as H antigens. Another example here is Escherichia coli, O157. Again, the H antigen is H7. Again, helps to identify that form of E. coli from other serovars. Again, an example of the various types of motile cells. Axial filaments. Spirochetes move by means of axial filaments, or an endoflagellum. bundles of fibrils that arise near the cell's poles beneath the outer sheath and wrap in a spiral fashion around the cell. These can cause the spirochetes to move in a corkscrew manner. Endoflagellum and spirochetes anchored at one end of the cell rotate and cause the cells to move. You can see the difference, and I went through that rather quickly, of the axial filaments in the endoflagellum. Now previously with the you saw the flagellum outside the cell wall. Now you see the axial filaments actually inside of the cell. Many bacterial cells have numerous hair-like appendages referred to as fimbriae. They are shorter than the flagella and consist of a protein called pilin. They help the cell to adhere to the surface, such as mucous membranes, often a factor in pathogenicity. Pili are longer than fimbriae, the number only 1 to 2 per cell. These are sometimes called sex pili because they can function in the transfer of DNA from one cell to another. Fimbriae allow attachment. Pili are used to transfer DNA from one cell to the other. Here's an electron microscopy of a bacterial cell with pili. The bacterial cell wall is a rigid or a semi-rigid structure giving the characteristic shape of the cell. Cell walls can prevent osmotic lysis. They are made of peptidoglycan in most bacteria. The cell wall of a gram-positive bacteria is composed of peptidoglycan, which contains two sugars, N-acetylglucosamine and N-acetylmuramic acid. This is also chains of amino acids. You should be familiar with the two sugars that compose gram-positive organisms. The two sugars alternate with each other, forming a carbohydrate backbone. Peptide side chains of the 4 amino acid attached to the N-acetylmuramic acid are cross-linked to form the macromolecule of the cell wall. This illustration is out of your book demonstrating the polymer of disaccharide, the N-acetylglucosamine, and the N-acetylmuramic acid, linked by polypeptides. This again looks at the cell wall of a gram-positive cell. As you can see, the constituents, the teichoic acid, wall of teichoic acid, lipoteichoic acid, the peptidoglycan wall, the plasma membrane, similar to what we saw in the human system, again with the phospholipid bilayer, proteins that are either integral proteins or transmural proteins. And then we compare that with the gram-negative cell wall. You see lipopolysaccharides, you see the outer cell membrane, the plasma membrane, and the proteins in the cell membrane. Gram-positive versus gram-negative cell walls. You should be familiar with this slide. Gram-positives have thick peptidoglycan, teichoic acid. In acid-fast cells, it also contains mycolic acid. Acid-fast cells, for an example, would be Mycobacterium tuberculosis. Gram-negative cell walls, thin peptidoglycan, no teichoic acid in the outer membrane. Many gram-positive bacteria also contain teichoic acid. The cell walls of acid-fast bacteria, otherwise considered to be gram-positive, consist of peptidoglycan and waxy lipid referred to as mycolic acid. Teichoic acid, lipoteichoic acid, links to the plasma membrane. The wall of teichoic acid links to peptidoglycan. Many regulate the movement of cations. Polysaccharides provide an antigenic variation. The cell walls of gram-negative bacteria also contain peptidoglycan, but only a thin layer. These cells have lipoproteins, lipopolysaccharides, or LPS, and a phospholipid outer membrane surrounding their peptidoglycan layer. A periplasma, or a fluid-filled space, is found between the outer membrane and the plasma membrane. The outer membrane also provides resistance to phagocytosis, and the action or complement also a part of the host defenses. When the cell disintegrates in the host's bloodstream, the cell portion of the lipid A is released as an endotoxin that can cause illness. Materials may penetrate the outer membrane through channels called porins. Lipopolysaccharide, lipoproteins, phospholipids form periplasma between the outer membrane and the plasma membrane. Protection from phagocytes, complements, and antibiotics. O polysaccharide antigens such as E. Escherichia coli 015787. Lipid A is an endotoxin. Porins, proteins, form the channel through the membrane. Gram-negative outer membrane is shown to you in the previous illustration. Gram stain mechanism. So now, very important to understand the difference between gram-positive and gram-negative cell walls when we talk about bacteria. And this is where the gram stain comes in. And again, those of you who have already performed your lab can understand and appreciate why this is important. Crystal violet iodine crystal forms in the cell. Gram-positive, alcohol dehydrates the polypeptide. The crystal violet 1 crystals do not leave. Thus, in gram-negative, alcohol dissolves the outer membrane and leaves whole in the peptidoglycan, and thus the crystal violet can wash out. Leaves a residual color or a stain that looks reddish, which would be the gram-negative. The gram-positive, because it holds on to the violet stain, the crystal violet iodine stain, because it does not leave, it has a darker purpler color. Differences in gram-positive and gram-negative stain. Mycoplasma bacteria do not have cell walls. They are unique because they have sterols on the plasma membrane. Archaea do not have peptidoglycans in their cell wall. They have similar substances, a pseudomurin. Some bacteria of the genus Proteus may spontaneously or in response to penicillin lose their cell walls and become L-form. Later, they spontaneously revert to cell wall bacteria. Atypical cell walls like mycoplasma lack cell walls. Steroids in plasma membranes, archaea, are wallless. Walls of pseudomembran lack the diamino acids. Now, damages to cell walls. Lysozymes, which are basically enzymes in tears or mucus and saliva, damages cell walls in many gram-positive bacteria. If you recall, again in A&P1, we went into detail about intracellular organelles. We talked about lysosomes being a very strong digestive enzyme that was inside of vacuoles that were created inside the cell to help destroy or damage foreign bodies or substances that enter into the cell. So lysozymes as an enzyme can occur in tears and mucus and saliva, and they damage the cell walls of many gram-positive bacteria. A bacteria that has lost its cell wall is surrounded only by plasma membrane and the protoplast. Gram-negative cells treated with lysozyme retain much of the outer membrane layer and is called a spheroblast. Both are sensitive to rupture by osmotic lysis. Lysozymes digest disaccharides and peptoglycans. Penicillin inhibits the peptide bridges in peptidoglycan and thus will destroy the cell walls. Protoplast is a wall-less cell. Spheroplast is a wall-less gram-positive cell. L-forms are wall-less cells that swell to irregular shapes, and protoplasts and spheroblasts are susceptible to osmotic lysis. Plasma cytoplasmic membrane. Plasma cytoplasmic membrane is just an internal to the cell wall and encloses the cytoplasm. In prokaryotes, it consists primarily of phospholipids and proteins. Eukaryotic plasma membranes also contain sterols, making them more rigid. Both prokaryotes and eukaryotic membranes have a two-layered structure, molecules in parallel rows called phospholipid bilayer. Those of you who have had A&P1 are familiar with this term. One end of the phosphate is the water-soluble, the other is the hydrocarbon or insoluble. The water-soluble end are on the outside of the bilayer. The protein molecules are embedded into the membrane along with the phospholipids. They move freely within the membrane. This arrangement is called the fluid mosaic model. This is an illustration to your left, and then the electron microscopy of a plasma membrane. Phospholipid bilayer is the primary constituent, and many of you are already familiar with that term. This is the plasma membrane illustration. Again, the phospholipid bilayer, the peripheral proteins, the integral proteins, transmural proteins. Again, most of you are already familiar with that. If not, you should go back to your A&P1 and read, and you'll see that the chapter is very similar to as described here. Membrane is viscous as olive oil. When we talk about the fluid mosaic model, proteins move to function, phospholipids rotate and move laterally. The most important function of the plasma membrane is that it's a selective barrier. It is selectively permeable, semi-permeable, and contains molecules and ions that pass through whereas others do not. Several factors affect the permeability. Large molecules such as proteins cannot pass. Smaller molecules such as amino acids and simple sugars can pass if uncharged. The phosphate end of the bilipid layer is charged. Lipid-soluble substances, because of phospholipid content, pass more easily. Plasma membranes contain enzymes that help break down nutrients and produce more energy. The chromophores, or thylakoids, which contain pigments and enzymes for bacterial photosynthesis, are found in the plasma membrane. Mesosomes are folds in the plasma membrane that may be only an artifact or preparation for electron microscopy.